So now we are we welcome Eugenio Cinquemani from INRIA Grenoble, who will talk about inference of statistics of a promoter process from population snapshot gene expression data.
So, well, thank you very much for the invitation, for being here. And I thank you very much, Ulis, for his presentation, which was actually very nice. And actually, it is also nice that it came together with mine because it has a lot in common in the sense of like the context. I was partially scooped because this is going to be about fluorescent reporters indeed, but this is all good. So it's okay, let's start. So okay, so I will introduce what fluorescent reporters are about. It's a technique to monitor gene expression dynamically, even in single cells. And then I will basically move on to the modeling of single cell gene expression, which is what you have already seen in part with Ulis, and a commonly used tool, which is called the moment equations. And then eventually I will use this setup to tackle an inference problem where what I want to infer is what drives gene expression, but not in terms of a deterministic signal, but rather in terms of the statistics of activation. And you will see what that means and how it differs relative to current work. Right, and then I will draw some conclusions. So let's start. So gene expression, you've seen it already. I illustrated here in a different sauce. So what I'm representing here is essentially the central dogma. So you have that upon activation of the promoter of a gene, which typically occurs when some regulating factor binds on the target promoter sequence, you get synthesis of the mRNA that the gene encodes, and then this mRNA, every mRNA molecule may give rise through so-called translation to several molecules of the protein it corresponds to. And these proteins, as you saw before in the context of gene regulatory networks, may itself perform some task, including it regulates metabolism, it may be building blocks for the cell, it may as well regulate other genes. So essentially, often people would like to understand in certain experimental scenarios how gene gets activated, okay? So dynamically over time. So one technique to try to look at this is the use of so-called fluorescent reporters, which means that basically it's a, first of all, an engineering approach, cellular engineering approach, whereby you attach to the same gene or an identical copy of that gene of interest. You recode what's being produced from that gene. And what you code in the DNA is a sequence for a fluorescent protein. Such that, so here the assumption is that the target, the promoter of the gene of interest and of the reporter that we synthesize in cells is either just the same promoter or an identical copy of it. So the assumption is that we are essentially looking at the same promoter dynamics via a fluorescent reporter, because these fluorescent synthesized proteins can be quantified in time-lapse experiments. So the idea is you would like to learn how the promoter is behaving in a given experiment over time from measurements of fluorescence levels. Like here in this picture, I'm not also including the fact that mRNAs and proteins can also degrade. This will come later in the mathematical model. So to give an idea of what you can see in an experiment, this is an experiment in yeast cells. So these yellow spots at a given point in time, this is an image of yeast cells where we have encoded fluorescent reporters, yellow fluorescent reporters in this case. And this is just typically in video microscopy, this would be one image in a sequence of images representing a movie of how fluorescence evolves in different cells. Now the cells, of course, they replicate. There's a lot things to say, but the short story here is that if you look at different cells and you look at how the fluorescence evolves in different cells, you get curves, time-lapse profiles of this type. I don't know if you see my pointer. I guess you do. So every curve with a different color here you see is basically a different cell, and it's the evolution of fluorescence in that cell. You can see that these curves are very noisy. So all these cells are exposed to the same environment, but the red bars down here are stimuli applied to the cells, to the environment at different times. And so every cell has its own story. Cells are genetically identical, and however, you see dynamical fluctuations over time within cells. And also you see cell-to-cell variability, for instance, like the typical levels, if you can call them the plateau levels of this fluorescence, are different from cell to cell. So these are different types of noise. To go a bit more into the details, noise in cells has been classified experimentally and then also mathematically in terms of intrinsic and extrinsic noise as far as gene expression goes. Now, there are a very celebrated experiment here by Elovitz and company that shows experimentally what intrinsic and extrinsic noise means. Essentially, in very simple terms, intrinsic noise is whatever stochasticity takes place at the level of transcription and translation of a gene. It's like the randomness of these events. And extrinsic noise is basically whatever noise affects gene expression that it's not in the randomness of these events. Can be the cellular context, which is different from cell to cell. It can be a random promoter activation, can be other things. But so this is like two different sources of noise. Right. So we have to live with this when we look at single cell dynamics, and we can actually try to exploit this because the stochasticity that we observe may tell us something about how the system works, how gene regulation works in cells, as Ulisse also showed for gene network reconstruction. So here I'm really addressing a more basic problem, which I consider also as a potential prior step before we try to reconstruct interactions across genes. So here I'm really looking at the problem. Given data, given fluorescence data from the expression of fluorescent reporters for a gene of interest, how do you reconstruct information on the dynamics of the promoter that regulates that gene expression? And the data I'm looking at are in a way, they are at the other side of this trade-off that Ulisse was mentioning between having few cells with a lot of information or having many cells with less information. So what I'm looking at here are so-called population snapshot data. I think Ulisse called them in a slightly different way, but the concept is the same. Instead of having movies of this type, where you look single cells evolve over time, there are techniques, notably cytometry, which allow you to essentially, at different points in time, measure the distribution of fluorescence across thousands of cells. So many cells, but the cells you observe at different points in time are not the same. They are samples from a common population, but once you have observed them, you throw them away. So what you actually have in terms of data are distributions like this. At different points in time, you have different levels of fluorescence you observe in different cells for that gene of interest. And you have thousands, even 10,000s. So what you gain is the number of cells you look at. What you lose is the temporal connection between different samples. Clearly, the only way to relate these samples from time to time are their statistics, because the statistics are those of the population that you draw the samples from. So here is what I want to address as a problem. Given measurements of this type, reconstruct, in this stochastic context, reconstruct the dynamics of promoter activation in some sense. Now, the thing is that despite cells are exposed to the same stimuli, because of a random gene activation, random stochasticity, so random binding of regulators to the promoter, you can have that promoter activation is different from cell to cell. So it is in itself a stochastic process that is essentially taking a different outcome in different cells. And so what we want to reconstruct from this data, what we can aim at, is not individual cell promoter activation profiles, but it's rather the statistics of promoter activation over the whole population we are observing. And this is like the key point in which this work differs from many of the things that many works that have been done in terms of reconstructing promoter activation from gene expression profiles. Most often that is I mean, it's a very well understood and solved problem in a deterministic context, but here we are looking at the same problem but lifted into a stochastic context where the promoter activation itself is a stochastic process. Okay. Of course, I'm not saying that nothing along this line exists. I'm saying that this is less explored. Right. So So I just also forgot to mention that another line of work in this context is what I'm doing with Aline, where we assume single cell profiles being available. But I'm not speaking about that because it was not the right context and choice. So I'm presenting this this line of research, which is like, let's say, my personal line. Right. So let's speak about gene expression modeling. So I am essentially using the same model of Ulysse, which also goes under the name of random telegraph model. So random telegraph meaning you can see the promoter as the clicking of a telegraph. So it's clicked or unclicked when it's activated or not. This on-off behavior is modeled as some activation profile. The network that represents, so the chemical network that is used to represent this gene expression via transcription and translation is composed of 4 reactions. Transcription, which means from nothing, from just an active promoter, you get the synthesis of an mRNA molecule, denoted M here. The mRNA molecule itself may get degraded, or it may lead to the synthesis of one protein molecule. So from M, you get M plus P in a third reaction. Of course, this reaction can take place many times before that the mRNA molecule degrades. And then a fourth reaction that completes the model here is the degradation of the protein, this R4 here. So this 4 reaction model is captured by a stoichiometry matrix with 2 rows and 4 columns, 2 rows being the 2 species M and P, and the columns being the possible reactions. And for reaction rates, well, the first is the reaction rate for the transcription of an mRNA molecule when the promoter is active. So U is, let's say, non-zero. Then you get a new mRNA molecule with this at this rate. The second rate is for the degradation of mRNAs. The third is for synthesis of proteins. The fourth is for a degradation of proteins. Now, in the deterministic mindset, all of these are concentrations, and U is a deterministic profile. In the stochastic mindset we look at, all of these are random quantities, and the rates are rather propensities, that is, probabilities per unit time, to be a bit sloppy. And one thing to notice in this model, which is very important because it simplifies our life a lot, is that this model is in absence of as a function of some unknown input profile U. This is a linear model in the sense that the rates are given by a given matrix of coefficients, rate constants W, times the state, the current state X, plus whatever depends on this promoter activation profile, which is an input. Okay. So this is nice because with affine rates you can do many things. In Ulli's case, the network gives rise to a nonlinear model because U is replaced by functions of other states in a nonlinear fashion because it is where regulation occurs. But here we treat it as an input because we just want to learn about U. Neglect for the time being, not bothering with where that profile U is coming from. So in the stochastic context, one tool that is commonly used to study the evolution of the statistics of gene expression, in particular the statistics of fluorescence in a population of cells, is the so-called moment equations. That is, equations, in fact, differential equations that describe, moment equations that are deterministic equations that describe how the mean and the variance of the process evolve over time. And if you will, you also have the possibility to describe the dynamics of the autocovariance of the process X, where X is the, recall, the random vector consisting of mRNA and protein abundance. So the moment equations are very well known and easy to derive. And they look like this. It's a system of linear differential equations in the mean mu and in the covariance matrix sigma. And okay, the autocovariance function, I'm not looking so much here, but you can also describe that as a partial differential equation. And where essentially the input intervenes through, so the input meaning the promoter activation profile is embedded in this vector function f. For us, the vector function f is simply a vector which only has like one non-trivial entry, which is the promoter activity u. So all of this is very well known if u is some deterministic profile, so a given deterministic function. But for us, in this context, u, that is f, is actually a stochastic process because our promoter profile can take different outcomes in different cells. So how do we go about it? Well, the first step, which is what I did what I did published as part of a paper in Automatica a few years ago, is to extend these moment equations to the case where u, f, is a stochastic process with some given statistics. Here I'm considering second order statistics only. So the mean, the expectation of f over time, mu f t, its autocovariance function, and potentially also its correlation with the initial state of the system at time 0. So essentially what can be shown through a marginalization procedure is that if in absence of feedback, which is a tricky point, but essentially it means that you can condition on the possible outcomes of this promoter process u and the statistics of the rate functions. So the statistics of the network do not change. So under this assumption of absence of feedback, essentially the moment equations can be generalized as to include these blue terms that essentially account for the stochasticity brought about by the randomness of the promoter activation. So these terms are, interestingly, these are linear functionals, they are integrals of the statistics of the promoter process u. In particular, for simplicity, you can forget about this CF here. The most interesting thing here is that one of the blue terms here that enters in the variance of the evolution of the variance of the system state is a linear functional of the autocovariance function rho f of the promoter process. Okay. This is important because it enters linearly, and because actually we show that what really matters in terms of the resulting statistics of the system is the autocovariance function of the promoter process. So this is the tool essentially based on which I can now address an inference problem. The problem that I pose is, given a fluorescence snapshot data, which are essentially this type of data here. In particular, what I consider from this distribution is just their mean and their variance at different points in time. So I only look at second order moments. So given data of this type, which are time profiles of mean and variance like I have here in this figure, and assuming they are essentially some noisy version of the actual statistics of the cells, one can actually characterize approximately the level of noise you get in these measurements depending on how many cells you are observing. So given data of this type, the question that I pose is to now estimate the unknown mean and the unknown autocovariance function of the process U, because this is what I want to learn, the statistics of the process. Now, the mean is not especially interesting because it essentially boils down to matching the observed mean of the output with the observed mean, with the unknown mean of the input. This is a very simple problem that boils down to a deterministic deconvolution type problem. What is novel here is the reconstruction of the autocovariance function. Notably because the autocovariance function of this unknown promoter process potentially tells us much about the behavior of the promoter, because for instance, the decay time of the autocovariance function tells us about how long the memory of promoter activation lasts, for instance. And depending on the autocovariance function that you reconstruct, you may start hypothesizing different promoter regulation mechanisms. Okay, so really the interesting thing here is reconstructing the autocovariance of U. Now, because I'm having sample and noisy data, this problem is ill-posed because there's not a unique solution, but it's linear because, as we saw in the generalized moment equations here, the autocovariance function enters linearly this system of equations. Okay, this is something we can exploit. And okay, so it's a linear inversion problem in the domain of autocovariance functions, and we can solve it by setting up a Tikhonov type regularized estimation problem. So trying to fit the data essentially by imposing some degree of regularity of the reconstructed function. So how do I do this? Well, I don't want to go too much into the technical details here, also because more will come later. But the story is that you may assume the mean is known. It's a very simple problem. So the way I state the problem here is that is what we have here in this rectangle here. Essentially, I want to find the autocovariance function of U that minimizes the discrepancy between the solution of the model, the generalized moment equations, and the observed values of variance observed at different time points in the experiment. Plus a term here, which is a regularization term. It is what allows me to balance the data fitting with the degree of smoothness of the autocovariance function to be reconstructed. This is the classical statement of a Tikhonov type regularization problem. The important point here is that for the time being, I am making the hypothesis at this stage that the autocovariance function that I'm trying to reconstruct is a stationary one. Okay, so it's just function of one time variable. And of course, to be consistent here, I have to solve this regularized least squares problem, subject to the so within the space of autocovariance functions. And because the space of autocovariance functions is a cone, actually, this is a convex problem because it has like a convex objective function to be solved within a convex domain. Now, it's an infinite dimensional problem the way I stated it here because I'm trying to reconstruct an entire function of a continuous time variable. But actually, the way you can solve it, this one is by using finite expansions. You can pretend that the unknown autocovariance function is a convex combination of some generating functions, and essentially you can reduce the problem to estimating the coefficients defining this convex combination. So this can be brought down to a finite dimensional optimization problem, which is quadratic and linear. So it's an LQP problem. You can also preserve the constraints on the function to be reconstructed being a positive semi-definite function as the covariance function should be. Okay, so this is the way I state the problem. So basically, when you expand your unknown autocovariance function in a finite basis, let's say basis, over a certain number of generating functions, what you have to do actually is to solve for every generating function the generalized moment equations, because that is what allows you to then fit the data and to find the weights of the corresponding generating functions. So fine, I will show you what this gives from the data that I have here on the left for a simulated case study of a promoter process U being a binary process of the type Ulisse showed, on the type I called random telegraph model. So this is a process U that switches between 0 and 1 with some switching rates. What you get as a reconstruction of the autocovariance function of U is on the right. So the blue is the true, and the dashed lines you see in reddish type colors are the estimates, which are essentially unbiased. These are the statistics of the estimates I get over some Monte Carlo simulation of the estimation problem. And shaded regions are the uncertainty bounds around this. So essentially things work very nice, except at the extreme tails of these autocovariance functions, because there essentially there's very little data that enters the estimation of those times. And you may wonder why the data on the left looks so nice and little noisy. Well, this is because in fact these are profiles on mean and variance from samples of thousands of cells. So even if different cells individually fluctuate a lot, when you have a large sample, the noise on the statistics is that you can draw from your large samples is actually very limited, theoretically speaking. Now experiments in biology are always less nice, but this is the theory. So the considerations so far are that fine, we have analytical tools to describe our expression process when the promoter is itself a stochastic process. We have a method to reconstruct the statistics of this process from snapshot fluorescence data, as long as these statistics are stationary. The thing is, the stationarity assumption is strong, because in dynamical experiments, typically you have stimuli that change over time, and that typically affect the statistics of promoter activation over time. So the stationarity assumption is possibly too strong. Ideally, we would like to be able to reconstruct non-stationary statistics from this snapshot data. But this is a very complicated problem in general, because a non-stationary autocovariance function is a function of two time variables. So it's like a surface that you want to reconstruct. And from the data we have here, you only have data collected at time samples, which constitute essentially one-dimensional profiles. So you are trying to reconstruct a two-dimensional function from a one-dimensional profile, which is generally, like, even by the dimensionality of the problem, it's unbearable. So the question is, when can you do this? There is one case that I find interesting, and actually that I have relevance to real experiments, which is that of modulated processes. At least this is the term that I found as a possible term for what I'm talking about. So, which is like a work that I would like to finalize on real data. I actually know a paper, have the data. It's really a matter of trying to do it and see how it goes. So what am I calling a modulated promoter process? Well, essentially, our process before was F. F is the vector that contains the promoter activity process. And what I'm calling modulated process, especially with reference to control type experiments, F is a product of a known deterministic function G times an unknown process E. So you may think that E encodes the stochasticity of promoter, the inherent in promoter activity, and G is the way you externally modulate the level of activity of the promoter. Okay, so if G is 0, you can think you have put your cells in conditions where this gene cannot be activated. If G is large, for instance, like you throw into your culture a chemical that activates the response of that gene, then the promoter can start getting activated with stochastic dynamics given by E. So in this case, if we look at the statistics of a modulated process of this type, well, notably, you will have a time-varying mean, and in particular, you will have an autocovariance function, which is, of course, non-stationary, but it can be expressed as the product of G on the left and the right times the autocovariance function of E. And let's say you may as well assume that E itself is stationary, because now the time varying nature of the expression process is captured by this function G, which is a deterministic one. So essentially, the autocovariance function, so the non-stationary statistics of the promoter process F, are related to, in a known manner, to the statistics, to some stationary statistics, which are unknown, but they are more reasonably modeled as stationary. So essentially now, if you look at processes of this type, the inference problem remains the problem of reconstructing row F, which is now non-stationary, but clearly it can be formulated as the problem of estimating the stationary statistics row E, because G is a deterministic function that you may assume that to be known, especially if this is a control experiment where you know what you are doing, essentially. Fine. So what is the technical complexity? How does the problem change? Well, essentially, the problem changes in the computation of how these statistics enter your generalized moment equations. If you remember, I go back. So the autocovariance function of our unknown process enter in these blue terms here in a linear manner. This is still true. But now the terms that I have to calculate are more complex than they were before, because so the term that I have to calculate is essentially an integral of this type. It's an integral that also involves the modulating function g of t, which can be complex. So essentially, in view of using a finite basis expansion for the approximation of the covariance function of that I'm looking for, I have to solve this integral for some convenient choice of this basis functions rho. This I'm free to choose, and before that was allowing me to compute this integral explicitly, because this g term here was not on the way, it was not there. Now, no matter the choice of my approximation of the covariance function, I have these g terms on the way that complicate the calculation of this integral. So how do we go about it? Well, there is one case where calculating that integral is especially easy. It's accessible. It is where this g, the modulating function g, is either itself a spline function, or it can be approximated as such. And spline functions are very good approximators. So essentially, I can always make the hypothesis that the deterministic known function g is a spline. So the idea is that the integral can be written in this form here as g times h, where h I define it more generally in terms of the derivatives of g. And maybe you already see where I'm heading, because the derivatives of g, because this is a spline, eventually these derivatives will become constants, piecewise constants, over a partitioning of the time period, and then vanish for higher order derivatives. So without going too much into the details of this, the story is that calculating this, in the case of splines, calculating this integral can be formulated as in terms of the solution of an ODE system of degree d, where d is the degree of the splines that represent your known function g. So if you see the term h0, which is the integral of interest, its derivative over time depends on the term h of degree I plus 1, all the way to the maximal degree of the splines you consider, and then that constitutes a closed system of ODs of order d. We can exploit this to calculate for any fixed covariance function. We can calculate the solution of our generalized moment equation, so the variance of the output that specific autocovariance function would give, as the solution of an augmented system of equations, which comprises the original equation for the variance of your state as a function of the unknown integral you were after, h0. In turn, h0 obeys the system of ordinary differential equations corresponding to the fact that our g is a spline. And so essentially, if you solve, you can now use this augmented system of equations to calculate very efficiently the contribution to the variance of the state that comes from one specific choice of autocovariance function of your process. That allows us, once again, to set up a regularized least squares problem, very similar to before, where again, what you try to do is to fit your variance data observed at different time points, tk, with essentially the contributions with your unknown with the contribution from your unknown covariance function. And because we are using a finite expansion of your unknown autocovariance function, this corresponds to a matrix V at time tk times C. C are the unknown parameters corresponding to the weights that you need to estimate to reconstruct your covariance function, times this matrix V. V is simply the contribution of the different generating functions in your autocovariance space to the variance data that you have in from your experiment, plus, as usual, a regularization term, which is weighted by an unknown parameter lambda, which is the regularization factor that can be also estimated automatically from the data. I'm not going to tell the story. So this is, I hope the idea at least is clear. It's a bit technical, but now here are the results, which probably are the most interesting part. So what I'm doing again in simulation is now I'm simulating a modulated process. I'm simulating a modulated process. So E, the factor E is still a stationary process times the modulating process G, which is essentially the way I know my experiment is acting on cells in terms of activation of the promoter. So, and I am considering, just to explore performance, two different profiles of the modulating function G. One in black is 0 at the beginning of experiment and increases, and one is in red. It is active at the beginning and then it decreases. Of course, what I am simulating is the same stationary autocovariance function of factor E, but the data that I get are different because the modulating process is different. So if I look at the reconstruction of the autocovariance function of factor E, you see that what I obtain is similar to before. Now, essentially here, the red regions correspond to the uncertainty in the estimation when I have the red modulating function G. And in blue, it is the estimation uncertainty of the true autocovariance function that when I use the modulating function in black, the one that increases. So essentially, you see that in the tail, there is more uncertainty and there is significant uncertainty in the estimates. There's more uncertainty for one of the two modulating functions, which actually shows that depending the way I stimulate cells, I can learn more or less the statistics of promoter activation. For the rest, estimation is quite well behaved, except at the tails. What is now interesting to look at is the corresponding reconstruction of the statistics of the non-stationary product G times E, which is actually the statistics of the promoter activation resulting not only from stochasticity, but also from the fact that I'm actually controlling it, regulating it. And so you see, this is a two-dimensional function. Here it is depicted in color. And in black, you see the corresponding, a quantification of the corresponding estimation error. So what's interesting to notice here is that essentially the estimation error is comparably small when you're not at the tails. But in the case of reconstruction of the non-stationary statistics, even the tails are not affected by so much estimation error, contrary to the reconstruction of the stationary statistics of the stochastic process that drives gene expression. This is because there is this G function on the way that essentially couples the stationary statistics and the resulting non-stationary statistics in a way that essentially flattens down the relevance of the tails of the autocovariance function. So the story here is that the modulating function plays a significant role. So how you stimulate gene expression will play a significant role in what you can reconstruct from your data. Okay, so this is what I've done along this line of research. Let's say I developed the tools to analyze gene expression systems subject to stochastic promoter activation. I developed the methods to actually do this inference in stationary and non-stationary conditions. I could explore a bit the importance of the modulating function, showing that, so this modulating function is something that really pertains the way you do experiments on cells. So how you stimulate your cells to explore their gene expression response. And what remains to be done here really is to try that out on real data. Once again, data and scenarios where this specific problem and approach can be applied exist. It is just a matter of trying it out. And I'm notably thinking of the work of Jakob Ruus in Paris, who has data, this type of data for a gene expression system, which is precisely modeled as a modulated process. So I would really like to try that out, and I'm curious to see what we can learn about it. I hope it was not too technical, and I thank you very much, and I take your questions.
Thank you, Eugenio. Okay, I see Olivier had a question.
Hi, Eugenio. Yeah, I have a few questions.
Yes.
Maybe the first one would be, you showed live recording first, and then you move to snapshot data. And then my question was, why not using those live recordings as snapshot data? Because you can just make slices on them. Why do you have to generate a new set of data for that?
Yes, this was just for illustration purposes. The one on video microscopy. I mean, it was to illustrate a bit the stochasticity of gene expression. Except that Ulysse had done it for me before. But but it's good because the point is precisely this. What Ulysse mentioned. You have a trade-off in video microscopy. You have less cells, and you have to have a video, a data process, and a non-trivial data processing step. You have the you have the tracking, segmentation, whatever. In cytometry, you don't have the temporal information directly, but you have tons of cells and zero processing essentially. This work is in the mindset that you should develop methodologies adapted to the different experimental possibilities, because different experiments have pros and cons.
Sure, I fully understand that. Maybe the other question related to that is, how many cells do you need to apply your algorithm on those data? I mean, is this, can you go for hundreds? You have to go for thousands, or?
Yes. So here, the simulations I did here. Here I simulated. I really simulated with like Gillespie type thing. I simulated 10,000 cells.
Okay.
And I actually simulated more. I actually simulated 100,000, and then at different points in time, I picked a subset of them just to try to break the correlations across time points, because because in cytometry this is the case. Your samples are much smaller than the population. But let's say in cytometry, taking let's say taking every 5, and here I simulated data taken every 5-10 minutes, which is like what you can do in cytometry, and counting, that is quantifying fluorescence for 1,000 cells in cytometry takes less than a minute.
Yeah, it's really easy. No, that's
if you want, no, but just also for the others, if you instead want to go, let's say that what I'm simulating here, having 10,000 cells in every sample, is doable. It takes every sample would take like 3-4 minutes, I guess, in cytometry. It's doable. It's in the range of the things you can do, let's say.
Sure. If I may have another question.
Yes.
David Sutter, in his luciferase paper, made the point that the half-life of the reporter was important the shorter the half-life, the closer you get to the promoter dynamics. So would that be also the case in your setting? Did you try like to lower the half-life of fluorescence reporters, or?
So I'm very happy of this question. Here, I'm assuming that the reporter is a known system, meaning I'm assuming that the rate constants of the reporter system are known. They may, but let's say reporter systems typically you characterize them before the experiments. So well, you can characterize them before usage. But it is true that parameters are not always known precisely. So the question becomes, can you estimate them? Well, one possible extension of this work is to try to simultaneously reconstruct the promoter profile and possibly also some partially known values of these rate parameters. I'm not sure to what extent this is doable, honestly, because the two things may conflict. But there's another line of work which I haven't finalized yet, which I really one day I want to do. This generalized moment equations, you can use them to entirely characterize the gene reporting system in terms of spectral properties. That is, essentially characterize, depending on the choice of the parameters of your reporter system, what kind of information the reporter system is able to convey, and what kind of information you lose because it gets blurred into noise, which is really like a mathematical formulation of the question: should we take destabilized reporters that decay very fast, or not? Because destabilized reporters typically are able to report on the details of the fluctuations, but they give less intense signals that may be blurred by noise. So you can study that trade-off.
That's a trade-off. Okay, thanks a lot.
You're welcome.
Are there other questions? Yes, Ulysse.
Yes, so thank you for the talk. I have two little questions. Maybe a first one which is naive, but I was wondering. So the goal is to reconstruct an autocovariance, and because you say that you use snapshot data, and so as you say, the snapshots break the trajectories, and for me it kind of sound magic that you are able to reconstruct autocovariance using only snapshots. So can you explain?
But research is magic.
Ah.
No. I'm kidding. It is. Yes. Let's say I'm looking at autocovariance because the problem of reconstructing means is easy. So it is also intuitive to understand why the mean the mean of the promoter process will reflect in the mean of the fluorescence function. It is not equally easy to have a grasp why the autocovariance function reflects into the variance data. But the intuition is the following. If I look at, let me see if I go. Indeed, before I developed the math, it wasn't obvious at all to me how the statistics of the promoter would enter the statistics of my system. Now I was hoping to have wait here. So what matters is the in the data here, the red crosses represent the evolution of the variance. So what is the evolution of the variance? The evolution of the variance is the evolution of the extent to which cells differ among themselves, to what extent the cells get dispersed over time. And this variance changes over time. So at the beginning, all cells here have the same fluorescence level, and then as time goes by, they get more and more dispersed. essentially, what the autocovariance function of the promoter does is to influence the speed at which this variability builds up. If you have a promoter process with an autocovariance function which is very peaked in 0, that means that the promoter process is essentially random noise, entirely random noise. Which means that this random noise instantaneously, because it's completely random, so very fast, it would make the dispersion across cells increase over time. So you would see a curve here in red that would ramp up much faster. If, on the other hand, you are in the opposite case, where the promoter process has a long memory, that means a very flat autocovariance function, which also means that when it gets activated, for instance, it does so for a long time and in a very and also rather uniformly across cells. Then in that case, the cells will follow similar patterns, and the dispersion of the gene expression level of the different cells will not increase as fast because they will all follow similar patterns.
Okay, thank you.
I hope it's a tricky
no,
part.
but it's much more clear now. Thank you.
Okay. You're welcome.
Can I have just a second little question?
Yes, of course.
So yeah, because I'm not a specialist of reporter technique, but I guess that the reporter molecule is a protein.
Yes,
right?
it is.
So because you want to reconstruct the promoter dynamics,
yes,
given protein.
Yes.
And I was just wondering if it's possible to how to say that, to go much closer to the promoter, like reporter mRNA. Would it be possible to get that? Or?
Yes. Well, yes. Here. Yes, indeed. This work is targeted to fluorescent reporters. So there you quantify things by light. You don't really look at the protein molecules, but rather the light they emit. If you have mRNA-seq data, for instance, as Ulysses showed, I guess you could do something similar. As long as you, yes, I mean, you could do pretty much the same thing as long as this data gives you, let's say, histograms of fluorescent reporter concentration, mRNA concentrations at different points in time, you can apply something similar.
Okay.
The only thing you need is a decent model of how promoter activity relates with the mRNA levels you observe.
Mm-hmm. Okay. Thanks.
You're welcome.
I have a question on the difference in the quality of the estimation that you have for different function G. So this is very interesting. And do you have an explanation for why this one?
My answer is the following. If you look at my paper, you will find the story that I made up about it.
Okay.
But honestly, it is quite tricky to understand. My best qualitatively speaking, my best explanation of this is that for G being the black curve, G is 0 in correspondence of where you have most data on the transient dynamics of the system. So somehow, when G is small, you have less information, and this information is where the system would give you most information, but you're kind of like canceling it out because of the shape of G. This is my best understanding of it. I think I tried to dig a little bit further into it. I to be honest, I don't remember if at that specific time I had yet another additional explanation. It's something to be investigated. Indeed, it's important because it's about essentially experiment design, how you want to drive cells.
Yes. But it may also be because G is always bigger. The red function is it's not always bigger, but it's
mostly bigger. Yes.
Yeah.
Did
you try with a G function with the same shape as the red one, but
no.
Smaller values, yes, or decreasing faster? Yes. Okay.
No. I mean, I tried a few, but I wanted to have two decently comparable choices of G, but qualitatively different, to see if I observed different behaviors. I didn't investigate further. Like, there's a lot of work that could be done here. It's just that it's my own and marginal research axis for now, unfortunately. Unfortunately.
Okay, and any chances that this dependence on G could be expressed analytically, or?
Look, all of this has to do with linear integral operators in the end, in a way or another. So I am sure there's a lot of analysis that can be done.
Yes, because as you said, it's related to optimal experimental design, and then maybe we want to optimize with respect to G. So
yes.
Yeah.
Yes.
Okay, thank you.
You're welcome.
Any more questions?
Yes.
Yes, I didn't know if you forgot to.
No.
Please go.
I re-raised my hand, but there's a
Okay.
question in the chat maybe first. Julien Chevalier asked a question.
Yes, Julien, you want to, or I can read it. Does the right plot on this slide is generated with the black GT curve or the red one?
I think it's with black, I would say. Good point. I don't remember. Yeah, I would say it's with the black. I think I got the same, pretty much the same story with any of the two, but I think the right one is plotted for the black one. Yes, but the story was the same.
Okay.
My question would be on practical matters. If you want to use a real live data, you have to infer the shape of the signal GT, because you said it depends on what you do to the cells, but it's more how what you do to the cells is transmitted down to the promoter, which might be quite different, right?
True. I didn't insist on this, but I don't even know if I have any reasonable information about it. Well, essentially, so here's the important relationship here, that my promoter process F is the product of G times E. E is the only stochastic part. E is a stationary factor. So the mean of F as a function of time is equal to G as a function of time times the mean of E, which is a constant. Okay, so essentially the mean of F is essentially the mean of G. Okay, so it's essentially given by G, okay, up to a constant factor.
Okay,
which means that from the mean data I can reconstruct G, because from the transient behavior of the data, of the mean data, so essentially the black profile here in the data, well, that response of the system is essentially given by G. If G is unknown, I can try to reconstruct it by the convolution approach, which is using only the relationships among the means. So in principle, G can be either reconstructed or refined from the mean data of the system. Now, to what extent this reconstruction is precise, well, it depends on the quality of the data,
yeah,
but in principle, it's possible.
Okay, cool, thanks.
You're welcome.
Okay. Any more questions? So to relate with the talk with Ulysse, here your promoter activity is independent of, it does not depend on the protein concentration. And it's an important assumption if I...
No, the promoter here, okay, the assumptions on my promoter process
Yes.
are, it can be any stochastic process with well-defined second order moments. It doesn't even need to be Markovian. The crucial assumption is, also the trickiest, is the absence of feedback in a stochastic sense. So essentially, when does this apply? Strictly speaking, for instance, my process U could be a result of the regulation from another gene. Why not? It's just the result of some other dynamics of the cell. This is fine, but this is fine. It still sticks with my assumptions, except if the expression of the gene I'm looking at feeds back into the activation of its own regulating gene by forming a loop. In this case, you can no longer say that stochastically speaking, there is absence of feedback, because there is feedback. So the output of my system feeds back into its input through a direct or indirect process. So if there are loops, like for instance, the case of bistable switch, strictly speaking, the assumption of absence of feedback is not true, strictly speaking. Now, how important that is, it is difficult to say, because if you have a very remote regulation path that comes back to the same promoter, maybe the input and the output essentially can be considered not in a causal relationship. I mean, you can, the fact that the output does not immediately regulate the same promoter may still be acceptable.
Okay.
And also, yeah, well, I take the chance to say that another reason why I'm looking at this data is because I consider this as a possible preliminary step, precisely to explore where the regulation of a promoter is coming from. Because if I reconstruct the statistics of the promoter from one experiment, then from another similar experiment where I look at another gene and its own statistics, I can try to relate the two and see what kind of model stands between one gene and the target regulated gene. So it's also a way to try to, a first step toward the gene expression, gene network reconstruction, but that part is not developed here.